athletic complex over there. And so you go whenever you get here. You need to be there. They say they're starting the test at 7:40. That's basically when they want you to sit down, and so they can start the test at 8. Really, so they're trying to get everybody there by 7:40 so they can get everything distributed and all that stuff. So I would say plan on arriving there a little before then. Maybe 7:35 or something, walking in the door there. And you need, hopefully that you need a pencil, you need a pen, whatever your test. Ours is both, so pencils and pens, and bring those with you. Bring like a bottle of water or some sort of water carrying device. Bring a snack because the test is long, though you can't bring any of that in the room with you. They have you set your little snack bag outside, so I would have it contained in a little bag, paper bag with your name on it, or a little lunch bag or something. So I'd put snacks in there. I would put like a drink, maybe some juice, like a little juice, something to kind of amp your energy halfway through, because it's like a marathon of testing is what it is. And so you want to get that second half just as energetic as the first half. So maybe just a little something. Protein is good, like maybe a breakfast bar, like a protein bar, something like that. So that you can just kind of keep your energy up, because that's kind of the hard part of the second one is maintaining your energy. Yeah.
Are we going to be marked absent in our classes when we take the?
Are you going to do what?
Be marked absent in our classes?
No, it's a student activity, but you have to be back like after fourth. Basically, they typically, and I'm not going to say always, because some of the tests are shorter than others. But they typically excuse you or have your student activity go through fourth hour, and the lunch is attached to it. That way they don't have to figure out who's which lunch. And so typically you have to be back by fifth hour. So, which is it is what it is. Like, it's not my favorite thing, because I kind of wish they'd just be like, hey, you've been testing, go ahead and go. But you will be counted absent for fifth, sixth, and seventh if you don't come back. Okay. But typically you have a student activity through fourth, and I'm sure they'll let you know if that's not the case. I know some of them that have a portfolio attached to it, like art and stuff like that, that maybe, I don't know if art has a test, but they have like a short test. You may not be excused all the way through fourth hour. So I'm not sure. But then again, not all tests start at 8 a.m. or 7:40. Some of them are afternoon tests. So make sure you are in charge of figuring out what time you need to be where. Okay. So if it's an afternoon test, you need to be there at the time it says. If it's a morning, be there at the time it says. So just be sure that you're paying attention to what time you're supposed to be at the doghouse. Okay. And so I'd say, after you have your snack, I prefer, if you have a little pencil bag, you can do it. If you're nervous about them not letting you have a one that you can't see through, they're not super strict about that, it doesn't seem like, but you can just do like a Ziploc bag with all your things in it that you need, like your pencils, your pens, eraser. And then in that bag, I would actually put a little packet of Kleenex, honestly, and maybe like a couple mints or something like that, just some stuff in case you start coughing, because you all know, like, once you start coughing, you're like, oh my gosh, everyone's looking at me, I can't stop coughing, and then you like have a coughing fit. So just things like that you can be comfortable for 2 90-minute testing sessions, to make yourself more just comfortable doing it, so that the focus will be on the test and not on all the environmental stresses and angst that can come with sitting in a room that's totally silent for 90 minutes and trying to be quiet the whole entire time. It gets just a little bit, like where you feel like everyone's looking at you if you make the least little noise. Same thing with Kleenexes. Every time you have to like raise your hand and ask someone to bring a Kleenex, it breaks your concentration and those around you. So it makes you feel bad, like I shouldn't ask for it. I might bring an extra mask if I were you, honestly. You have to wear your mask your whole time. I might stick one of the disposable ones in your little pencil bag too, just in case, maskadents, wherever you like sneeze out a whole entire who knows what into your mask, and you're like, well, that's the most disgusting thing that's ever happened to me. So you may want to bring an extra mask with you just in case you have a little accident in your mask. This is things we've never thought I would have to say ever in my whole entire life. But anyway, so basically then you'd sit for the first section. They let you have a break. Oh, cell phones are not allowed in the building. So with my students, I don't know how your teachers are going to do it, but with my students, I have like little breakfast in here for you guys, like 7:15, 7:10 or whatever. I'm in here and I've got breakfast and maybe some little waters and juice and things like that. And then I have you guys put all of your stuff in my storeroom. Like, your backpacks with your phones and everything in it. And then I lock that door until the test is over. And so that you can feel confident your stuff is secure and out of the doghouse, because you can't have your phone on break. It's just like the ACT. Like you can't have your phone anywhere. And so so that's what I do. I don't know what other teachers do because they don't have storerooms. So, I don't know what they do. But you may want to ask whatever teacher's a subject if you can leave your stuff in their classroom or whatever. If you're not comfortable, I'm not comfortable leaving my phone in the car, in the parking lot. But if you are, then that's fine. You can hide it somewhere or whatever. But definitely for my class anyway, on that test, that's an option. I mean, because I don't have anybody I have third hour basically is my first class at that point. So it's pretty quiet in here and your stuff is pretty secure. But anyway, let's see. Any questions about anything? They just kind of wanted us to go over with you kind of what the expectations are and make sure you don't have any questions about how the testing, because we're starting next week. So now sophomores, I don't think you guys have a test until the week following, but no, juniors and seniors are starting to have tests next week. So I guess I won't see you guys on Monday, and then Tuesday, what's Tuesday? Anybody have? Oh yeah, I should know. May the 4th be with you. Calculus is on May the 4th. And then Wednesday's English for seniors. So seniors get hit. Boom, boom, boom. 3 in a row. And then Thursday. We have something on Thursday. I can't remember what it was. Oh, it's A-Push.
A-Push is Friday, I think.
Is it Friday?
Yeah.
Okay. I thought A-Push was Thursday. Okay. So there's something coming up there. I know that I had quite a few students doing. So it just begins and we're on the downhill slope. Almost done. So that's good news. All this like worry and stress can be behind you. Won't that feel good? That'll feel really good. Okay. And then we can check 2020, 2021 off and be like, done. Let's move on with our lives. Let's be one of that, pretend like that one didn't exist. Okay. All right. Unit 4, cell communication. Let's see how long this takes. I'm not sure how long this is going to take. So we'll see. So this one's kind of weird. It's kind of disjointed. I typically do like one chunk here, basically maybe kind of thirds again. Actually, mine's off. I put my center off. Oh well. Kind of thirds again, except our thirds are going to be divided too. It's because I'm kind of making boxes of all the different topics, and it's strange. I'm trying to figure out how to connect it. I've drawn it like 3 times. I can't quite get it perfect, so we'll see. We'll see how I do. Okay, so cell communication is basically it encompasses how cells get a message and what happens to it on the inside of the cell. But it also does the cell cycle and its regulation. It also does feedback. And it just seems like this kind of mismatch of weird communication activities. So that's why it's a little bit difficult to get it all like concise. But the first thing, I'm going to leave a little space here underneath here. This is going to be like 4 little bullet points on what the response is. So do I want to do that? So I'm trying to figure out where to put stuff. That's my problem with this. Right in the center, which mine is not in the center. Right in the center, I kind of have a general. I keep trying to call it basic, which is why I drew a line there. But it's really just a general. So general signaling. So all signaling. The signaling has 2 L's, by the way. Should. Okay. So general now it says signaling. I like it. Okay. So general basic signaling they all kind of have the same basic parts, like 3 basic parts. Like you can number 1, here's what happens. Number 2, number 3. And then it's like all the rest of signaling is like variations on the theme, like something different happens in step 2, something different happens in step 3. And so the first thing you just really need to get on there, the basic things that happen with each one. I'm going to make this into a box eventually, but I'm not sure how big the box needs to be. So I'm going to just draw. So first of all, you always have to have something that sets it off. So I'm going to draw a little teardrop shaped thingamajigger here that I'm going to call a ligand. So ligand is a general term. Ligand can be a lot of different things. And so, for example, it can be a protein. It can be a chemical, just a small little chemical of some sort. It can be a hormone, which also, by the way, can be a protein, but is often a lipid. So a ligand can be a protein, a chemical, something. Okay, something is setting this off. So a ligand is what's going to set it off first. And so I'm going to draw a little cell here. I'm going to put a little dotted line like the ligand is going into the cell, or not into the cell, into the receptor. And then I'm going to draw my little cell membrane. Here's my little cell membrane. Okay, put a little gap in it for the protein. And I'm going to make a protein that fits the ligand. Okay, so here's just a protein. So that would be your like membrane protein. Okay, so step one, this is going to be your step one of this signaling, and that is the reception. Okay, so step one is reception. I don't know how you want to do that. And my one is kind of not right in front of my word, but step one is the ligand reception. So basically here comes the ligand. Here's a protein. And so this is a receptor. And it's a protein always. And it's in somehow embedded in the cell membrane. Now, of course, your reception protein can change kind of how it works. It can work in a lot of different ways. It can be a channel instead of just a protein. So, that's still a protein, just a different type of protein. So this is what I'm talking about. This is a general thing. So first you have to receive the signal, and then you're going to have a transduction of some sort. So that's going to be step two, you have to transduce it, or the signal has to go through a transduction, which is where it moves through the cell. Okay, so that's going to be step two. And then step three, I'm going to draw a nucleus in here, even though it doesn't have to be a nucleus. You need to erase all that. We'll just start over if you need to erase everything.
I just only wrote it in pen.
Oh, I was like, just get a new piece of paper and start over. Okay, so this is my DNA here, and I'm going to do, this is just a general example, right? Because it could be a lot of things. Do you remember what that arrow means, by the way? I wanted to add that because I wanted to make sure we go over kind of some arrows. Do you remember what that arrow means when you see it on a DNA strand like that? It's coding. Yeah, you're starting transcription, right? So I'm saying this is 3 here, 3, wherever I'm going to put it, and I'm going to call this the response, okay? And it doesn't have to be, and I don't know if you want to write this, if you think you're going to forget it, but it doesn't have to be transcription. Like response could be, that's what I'm going to write up here is the 4 different types of responses you can have. That's what's going to go up here. Because it's kind of like the end game of signaling. So I thought I'd put it right underneath the title. Basically, your responses can be a lot of different things. But in this case, it's like, okay, a ligand came along, locked into the protein, was somehow transduced to the cell, and number 3, you got a response out of it, okay? That's general signaling, step 1, step 2, step 3, okay? So sometimes in the past, I've seen them actually just ask that. Like, here is a general signaling, like label the steps. And you're like, okay, we have a reception, a transduction, and a response. I'm going to make a box around it. See, there we go. Okay, so there's your first thing, just generally how signaling works. Okay, and then we're going to talk, I'm going to take each piece of it kind of kind of give some variations of each piece. And then we'll talk about the cell cycle and feedback, and I think that'll be finished. So, all right, everybody got that part? Okay, so let's start at the end, actually. Like, what are the responses? So I'm going to start with number 3 here. So this is the response, and I'm just kind of keying what it is. So these are responses, and I'm going to have 4 of them right here. 1, 2, 3, see if I can fit them. Didn't quite leave myself very much room. So 4 bullet points on, and these are just examples. Okay, so like what kinds of things could you get from this? Well, one of them is you could get growth. So you could stimulate the cell cycle. Put that in parentheses. And
the cell cycle is like the mitosis.
It ends with mitosis. Yeah, G1S.
Interphase and then metaphase.
Interphase and then mitosis or meiosis, cytokinesis. Yeah, and we'll put the cell cycle over here in a second, or actually right here. But okay, so you could get growth. So you could say, okay, the cell's not going to grow until it gets an outside signal. That's true. It won't. Okay, you could get a secretion of molecules. Okay, so things like, now this is for your cells that produce stuff, like gastric enzymes, like different things. Like a lot of our cells, I mean, they're just kind of workhorses. They just produce things for our body. Being a multicellular organism, you have to have stuff to do things, and not every cell. We know that not every cell produces everything, right? That it's very differentiated. Different cells do different things. And so sometimes all you're doing with your ligand coming in here is saying, hey, we need some more melanin. We need some more gastric enzymes. We need some more, like this, that's what that ligand is doing. So secretion, whatever it's supposed to do. You might have gene expression or transcription. And if you want to put transcription in parentheses, you can, and you might have apoptosis, in which case that would be a lysosome. I'm just going to put lysosome in parentheses so you remember that's where that pathway would go. So if you did apoptosis pathway, you wouldn't go to the nucleus. You would go to a lysosome for number 3. If you did a growth, if you had a growth one come in, it would eventually end up in the nucleus because you have to replicate your DNA, but it would also probably be signaling organelles to divide and stuff like that, okay? Gene expression is going to go to the nucleus. I guess you could put nucleus on there if you wanted to as your final destination. And then secretion would just go, probably secretion is also going to go to the nucleus because you're probably going to be creating something, be a protein or something like that, some sort of enzyme.
Where's growth?
Growth, yeah. So growth will have a nuclear part because it has to the DNA has to be replicated, but it can also do like your organelle replication. Like, so it may be sending the signal throughout the cell to different things that need to go in. A lot of them end up in the nucleus actually because the DNA will kind of take control at that point. So because whenever it signals the DNA will make an mRNA that will go to the protein, and so then in that way, the DNA takes over kind of the cell signaling at that point. Okay, questions about that? Any questions? All right, let's talk about, let's see, what do I want to talk about? I don't think I want to talk about the ligand really at this point. I guess I can talk, I'm trying to figure out what, because I mean, we know that it's a protein, a chemical or hormone. I don't think there's anything else to say about a ligand. Anybody have any questions about ligand? Yeah.
Is it like the substrate?
I wouldn't call it a substrate just because I wouldn't say that this is an enzyme, but it's kind of the same idea, right? So it's like has to lock into that because in a substrate, usually the enzyme is acting upon it, like breaking it apart or something. And that's not the case here. In this case here, the ligand is locking in and actually causing some sort of change in the protein. And so substrate, we kind of leave that term with enzymes, okay? Ligand's a little bit different because it's a signaling molecule rather than something that's going to be broken down or something like that. Okay.
What are the 2 main points about a ligand?
2 main what?
What were the 2 main points of what a ligand?
I don't know if I said 2 or not. I don't remember. But anyway, what a ligand is it's some sort of signaling molecule that will be received by this protein. So it could be anything, honestly. Like it could be, you'll see a lot of different things used as ligands. But basically, it is a general term that means it's going to begin a signaling process. And ligand is just a very general term. Like it's not, this kind of, a ligand is a protein. No, it could be a protein, lipid, whatever. Yeah.
Whenever you have the thing where it's like a line and then another, like it's like a T.
Mm-hmm. Inhibition.
Is that like inhibiting?
Mm-hmm.
Or what is that?
Yeah, inhibiting. And we'll all have inhibiting lines up there when we do the cell cycle so that we can remember what they look like. Yeah. Okay, so let's talk about then let's do variations on a theme. That's what I'll do down here. It sounds like a violin piece. Okay, so let's see. I have 3 that I have, and I kind of go back and forth if I need 4. But basically, that's going to take up kind of this whole entire bottom here. We're going to do a G-coupled protein. And I'm just going to give you a little bit of information about each one, and then if you're like needing more, you may need to go like do a little more digging. But just to remember that they exist is kind of the point here. And then what was the third one that I decided was important? Okay. Okay, so these are actually somewhere, I think that we would consider a lot of these transduction, although a ligand-gated channel is actually kind of 1 and 2, like 1.5, sort of, because it is a reception and also passes it on. So so some differences. So these are differences, a little bit of maybe detail on your general signaling. So a G-coupled protein. This is one that has a G protein with it. I always say that backwards. It's actually probably technically G protein coupled reception. That's okay. It's coupled with a G protein, so I don't know why the G's over there, but whatever. Okay, so basically what you have here, I'm going to draw a little cell membrane, and then I'm going to put a little protein in it, just like this one. Okay, so it's going to look a lot like that one. So there's our receptor, right? So there it is, cell membrane. Okay, and so instead of just being like by itself, you have a G protein here. So I'm going to label that as a G protein. And if you want to fix it to say G protein coupled receptor, you can. I always call it G coupled protein, and then I sit there and go, that is totally probably not the right to say that. But I think they do say it that way, which is weird. No, it'd be fine. So this is a G protein, and it has some dealings with GTP. And so that's kind of why they call it that, though it's really difficult to find why they officially call it G protein, because it doesn't always have GTP involved. And so and I did some digging to try to figure out why in the world are we calling this a G protein. And it's like, it seems to not actually, we don't actually know very well. But anyway, so a G protein, the difference being here is that you will have a secondary kind of protein that is not a receptor protein, but it's still on the membrane that is activated by this. So a G protein, what happens is, this is why it's different from this one, okay? In this one, it's just like simple passing. This one, a G protein, will actually, when it's activated, it will then activate a molecule that is still on the membrane. So it didn't go in, right? It's going like across. And then this is going to activate a second messenger. And that's kind of the big deal with the G proteins, is they have a second messenger. It's usually C-amp. And that would be like we'd be activating it with this guy. So this guy's going to activate C-amp. Okay, and then it would be passed in. Okay, so that's going to be kind of the big difference with sorry, yeah, you can like stand up if you want. I know it gets really low really fast. My board just needs to be bigger. I need to be taller.
Does that say activation?
Activated. So C-amp would be activated by the secondary protein here, and then it goes in. So here's what you should know is that C-amp is a second messenger. Like label that because that is kind of something they like to pick on. I'm going to label it. Okay. So G protein coupled reactions or receptions typically have a second messenger, and that second messenger is typically C-amp. Though it can be other things, that is the one that they wrote in your standard. So I'm going to say you can stick with that. So why is the second messenger a thing? Okay, so basically, how do you identify it? It's not a protein, right? So all the other ones of these, it's going to be like a protein cascade, if you will, like pass from protein to protein to protein to protein. Okay. C-amp is not a protein, and so it's got a different role in the signaling thing. Now it's called the second messenger, which then starts the protein cascade. Okay, now that's not in every one. I mean, that's one type. Yeah.
So pretty much the G protein is the first messenger, and then C-amp is the second.
Yeah, but we have a friend here, and so that's kind of. You have something that has to activate the second messenger. And so that's kind of how you can tell that it's a G protein type of signaling pathway, is that it activates something that's also on the cell membrane, but not a receptor. So if I were you and you have a second, I'd like go Google a picture of it, like an image of a G protein coupled reception, because that kind of helps you to like say, okay, I see what we're talking about now. But really, it's just one type of signaling. So it's not going to be every time. Okay. Okay, so that's one. Amplification is the next term. These are kind of terms that I just kind of picked out some terms to show you what they are. Thank you very much. Okay. You have one. Oh, I have a lot of them. I thought there were just two. Unbind those here. All right. Yeah. Yeah. Okay. You guys must not have gone to the word assembly. Okay. All right, so for amplification is basically, hey, we got a signal, and now we're going to amplify it within the cell. Now, typically on this one, it's not so much you get a faster response, it's that you get more of a response. So it looks kind of weird. You can usually tell what I'm going to draw once again. There's my little signal. There's my little receptor. Starting to look like a tulip, and that's my little protein receptor. Okay. And then it's like going to come into here, and it'll have like a little cascade. So let's do another protein. So protein number 2. And then all of a sudden something, and this can be so broad. I almost don't even like teaching this one because it can be so broad. Something sets off an amplification. And you have, instead of it just going 1 to 1 to 1 to 1 to 1 like this, you all of a sudden have like a fan out of arrows. And it's setting off. So at the end of each of these arrows, it's kind of doing the same thing. Like, so you'd have like a bunch of proteins at the end that are identical. And so it's just setting off this huge chain of like a response. And so instead of 1 to 1 to 1 to 1, now all of a sudden you're getting 1 to 1 to 20, and you're getting a whole bunch of product. Not that the signal's moving any faster. It's just creating more of whatever it is. Okay, so anyway, that's amplification. It's really obvious when you see it because it looks like a fan of arrows, and that's kind of weird looking in a cell signaling pathway. So it's like, oh, it's being amplified. Signal's being amplified. That's kind of what's happening there. Just some vocab words, I guess, we're going to stick in here. All right, so then ligand-gated channels. These are kind of fun. All of a sudden, my tulip is going to have to be split in half, my little receptor protein. Okay, so in this case, we have a ligand, but the ligand, instead of just, okay, so hang on, let me draw first. I'm splitting my tulip in half. My tulip receptor, making it into a channel. Okay, so now it's a channel. Okay, so how this works, this little protein here, is typically it'll have what we call a conformational change, like it'll change shape on the inside. So the ligand doesn't get in, right? So the ligand doesn't get in, it just locks in, the protein goes, changes shape, and that shape change causes the next protein to shape change. Maybe even causes like a phosphorylation, like a phosphate being added or something, causes something. And this is the problem with cell communications. It's like, I don't know where they're going to go with this. Like they could go 100 different ways. So basically it's being able to just read the diagrams and figure out what's going on. But typically what you'll see in a typical signaling transduction is that the ligand locks on, does not enter the cell. The receptor protein changes shape, which kind of causes like a domino effect, like you pushed a domino. And then the next one goes, the next one goes, the next one goes, the next one goes. Okay. Ligand-gated channels, a little different. You have two options. The ligand can actually enter the cell a little bit. Usually it doesn't go far. Usually it'll just kind of go to the end and then start the chain reaction itself. Or it can lock onto the outside. So I'm going to put mine locking onto the outside. So here's my little ligand. Okay, so there's a little spot for it to lock into, a little ligand area. Okay, and when it does, then it opens and something else can enter, like calcium or something. Okay, so you could see something else enter on the inside. But the ligand, so there's your ligand. The ligand, it does the opening of the channel, which then starts the signal. Okay, or the ligand itself can enter, go halfway through, lock in somewhere inside, or go all the way to the end, lock in on the bottom. Or so you have a few options for your ligand here. It can just lock on the top, open the channel, and something else comes in and starts the signaling pathway. Or it can start the signaling pathway itself somewhere inside or something like that. Yep.
Are the ligands connected to the other two responses?
These two, yeah. Yeah, all of them have to begin with a ligand. All of them begin with a ligand. I'll draw a ligand in them. Okay. Okay, so that's a ligand-gated channel. Ligand-gated channel can also be closed. So maybe it's open and it's like, it's naturally normally open. The ligand hits and shuts the channel. whatever. And then maybe that starts off your signaling cascade. But just know that when this is the case right here, a ligand-gated channel, we're typically talking about a signaling pathway will be started and not just opening it for something to enter that, the cell needs or whatever. Okay. Not like osmosis or something, but actually starting a signal. Okay. So those are just some variations. You can see some vocab words that might pop up. Make sure you know second messengers are a non-protein, kind of mid area messenger, somewhere in the middle. Okay. All right. Questions on any of those terms? Any questions? Okay. All right. Let's talk about transduction a little bit. I already kind of did, but I'll write it down here for you. Okay, so you can have the transduction can be a protein. Oh, sorry. That means it's, I guess I'll write transduction there. Number 2 means it's transduction. So I try to kind of label them based on like what's going on here. So you can have a protein modifications that can keep the signal going. I can't spell today, I guess. So like shape changes, etc. Okay, or you can have phosphorylation, which you'll see because the little P will be connected or disconnected. So would be like, you'd see like that thing. So that's like a phosphate added or removed. Those are two of our kind of major ways that the signal keeps going. And that can cause a cascade. Do you know what I mean by cascade? Do I need to define cascade for you? Like cascade, like step by step by step, like a domino effect. So these can cause a cascade, or they can be a part of the cascade. So maybe that first protein changes shape, and then the next one changes shape, and the next one, and then you see a phosphorylation. All of a sudden, a PO4 is what it is, but it'll look like a P with a circle on it is how they'll designate it. All of a sudden, that's added, or then it's removed, and then so you're just seeing kind of some step by step by step by step by step. So this is going to just basically keep the signal going. Okay.
What was the word you used?
Cascade. All right. I didn't quite mean for that to go outside of that. That makes it look kind of ugly. Okay, so let's then do, what else did I have at the top? I had cells, so signaling can either be, so then I have kind of little boxes. I know I told you this is full of boxes, sorry. Your signaling can either be direct, so it can be cell to cell, or with a gap junction. So just a little space, or it can be distance. And that's when we're talking about hormones in bloodstream. For us, right? Obviously plants don't have blood, so they would be hormones carried in their cells. Plants do have hormones though. Okay, so this is how like kind of signaling can be done. So touching cell to cells, touching each other. So this could actually be, your little ligand could be a next door neighbor protein, right? It's sticking off of a cell membrane, and it's like, boop. And then when it touches, maybe it stops cell division, right? Because you don't need any more cells because they're touching. And then when it doesn't touch, comes apart, then your cell division might be, signal again, because now you have a space and you maybe need to fill it, something like that. It can be a gap junction like a neuron, where it's passing a neurotransmitter to the next cell, and when it picks it up, it keeps the signal going. Could be like that. Okay, a little space. It could be distance, and this is when. So how do I know it's a hormone? Because this was a question on the mock. How do I know it's a hormone? It was a multiple choice question. Basically, how I know it's a hormone is if it's created in one area and it causes a reaction in another area. So like in our multiple choice question, it said it's created in a bone, like the bone marrow creates the ligand, and then it's picked up in the pancreas. That's a hormone. That's not direct. That's not a gap. That's a long ways. So it's like that had to get in the bloodstream and go to the pancreas. So that's kind of how you know it's a hormone, because it is a distance. You have to find distance to be a hormone. Okay. Otherwise, if it's not going going a distance, it's not technically considered a hormone, because then it's cell to cell or gap junction. Okay. Question? All right. And then on this little side over here, we are going to have, let's see, my little paper. I swear I've drawn this a million times. Oh yeah, feedback. Feedback loops. I'm going to do a simple feedback loop. These are actually sometimes challenging to figure out what is going on, but kind of a rule of thumb is for a negative feedback. So we have negative feedback. I'm going to draw a little thing that's like a little loop. So plus, so that's increasing, and minus, that's decreasing. Let's use temperature for an example. So when the temperature is increasing, you start sweating, and then your temperature would go down. And then if it got down too much, you'd shiver. Okay, right? So this is basically maintaining. So let's just say maintain a set point. That's what it's doing. Okay, so your set point is supposed to be like your normal body temperature. And this could be for anything. My mask is itching my nose. This doesn't have to be temperature, obviously. It can be, let's see, on our mock, it was, actually, this was, on our mock, it was a positive feedback. It was insulin being released, and blood sugar, and then the insulin was creating more insulin, which was creating more insulin, anyway. So, but it could be a lot of different things. It doesn't have to be temperature. So, and then positive would be where you just see pluses all the time. Okay, so plus, 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 and this is amplify. That's what it's doing. Okay, so on this one, I like to use the example of ethylene. I'd like to use the example, but I can't spell it. Ethylene has a Y in it. What the heck, Miss Lloyd? It's not right. Give me a second. There, it's like that, I think. Maybe that E's there? I don't know, who cares? I think I have it written on my paper. What the heck? ETHEYLEN. Yeah, that's right. Okay, so ethylene is a hormone, and then I swear I cannot spell today. So then it creates ripening in fruit, which then releases more ethylene, which then creates ripening, which releases more ethylene, which creates ripening, with blah, 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 blah. And if I didn't spell that right, I mean, whatever. I'm not shocked. Okay, so on a positive feedback, you are going to be working with more and more and more and more and more. Like basically the production of the thing creates more of the thing, whatever it is. So like in our question on the mock, it was like you could look at the insulin pathway, and if insulin entered, then it went through this series of steps, kind of like signaling, and then what it ended up doing was releasing more insulin, which then went in and went through a series of steps and released more insulin, and then it was like, oh, that's a positive feedback loop. If it had been negative, it would have released more insulin, which would have created a series of things which would have stopped the production of insulin. Okay, so that's the difference. Is the product stopping the process of producing the product, right? Or is the product increasing it? And that's the difference, okay? All right, let's look at the cell cycle really quick. If you're not great with the cell cycle, HHMI cell cycle, we did it in class, or you did it at home as your remote work. That is the best resource I can find as far as figuring out cell cycle. So make sure you know, because some of you didn't. G1, S, G2, M, and then there's a cytokinesis at the end, but they don't really ever talk about that. It's kind of weird. They leave cytokinesis out. It's kind of sad. S is going to be your DNA replication. So DNA is copied in S. It asked you on the mock to put an X where something was occurring, and we didn't quite get that. You're supposed to put the X on S right there. Okay, and M is the division of the nucleus. So, by that definition, prokaryotes don't do mitosis, okay, because they don't have nucleus. So, this is only for eukaryotes. And then regulation of it, I'll put regulation right here at the bottom. Sorry for those of you who are, my table's in the way. So regulation of it, our two big regulators are P53, which inhibits, there's your line, Natalie. Looks like a little upside down T, which inhibits CDK, okay? Yeah, that's gonna be really low. So what that says right there, for those of you who can't see it, is P53 inhibits CDK. Okay, so CDK is actually in your standards. You're supposed to know what it does. So this is cyclin-dependent kinase. Remember, a kinase is something that cleaves. Kinase is a chopper. So kinase cleaves off a phosphate usually. So its job is basically to, and you know, because phosphorylation, the little P under transduction, you know that if you change the phosphates in one of these signaling pathways, it's going to change the signaling. So kinase is either going to stop something or start something depending on what the phosphorylation does in that pathway. So a kinase is one that does that, usually phosphorylates or dephosphorylates something. This one is just simply dependent on cycling. So this kinase depends on cyclin, okay? So if cyclin's not there, this kinase won't do its work, right? That's what that means, cyclin-dependent kinase. And they usually will write it out for you, although in our standards it says CDK. So cyclin-dependent kinase basically cyclin-dependent kinase will make it go. So this is go for the cell cycle, right? And P53 inhibits it, so it's stop. Okay, so cyclin-dependent kinase is on go as long as P53 isn't inhibiting it. Okay, so P53 isn't there to inhibit it, cyclin-dependent kinase will go. So your cell cycle will advance. We did, on this question, this was like, I don't know, the fourth question of the little, it was like, maybe, I think it was question number 4. Basically, it was either people got it about 4 out of 4, like all of the points, or it was not good at all, like 0 or 1 out of 4. And so, and it was all based really on if you talked about what CDK did, really. If you didn't mention CDK and you didn't mention that it made the cell cycle go on, then I couldn't give you points for it, because you had to say that the reason P53 was inhibiting the cell cycle was because it was stopping CDK from advancing it. And a lot of people just left that completely out. They like said P53 was doing something to the DNA or something like that. And they didn't talk about CDK at all. Okay, so that was kind of where we fell off on the wagon on that one. Okay, so make sure you know that's what CDK does. So CDK is the advancement on there. I think that might be it. Okay, questions on any of that stuff? I know it just feels like kind of and make sure you know what cancer is, right? Cancer is just the crazy growth. So cancer is CDK out of control because it's not stopping the cell cycle.